My thesis is on landscape genetics of salamander populations at Mammoth Cave National Park. The field of landscape genetics is a combination of two biological subdisciplines, that of landscape ecology and population genetics. Landscape genetics involves modeling the differences in allele frequencies between adjacent populations, while also accounting for the surrounding landscape features. My goal was to use landscape genetics to quantify both the genetic diversity of the population and the level to which the variation in habitat and barriers to movement affect that diversity. Amphibians are experiencing a period of global population decline. There are several proposed reasons for this, but the one that my project seeks to investigate is habitat fragmentation. The findings of my research will hopefully aid in the development of amphibian conservation strategies that will serve to enhance both habitat connectivity as well as genetic diversity for amphibian populations. The study landscape that I use consists of 50 sampled ponds in Mammoth Cave National Park containing Ambystomata opacum. I will seek to determine the best statistical model to describe the pattern of genetic structure between the salamander populations, and I will also aim to decide which vegetation types encourage and inhibit gene flow processes. Pictured here is the study organism Ambystomata opacum. It is commonly referred to as the marbled salamander, and it can be found throughout Mammoth Cave National Park. The hypothesis for my project are the isolation by resistance model would best explain the observed genetic diversity in the samples, and also that the vegetation type that will be the least resistant to gene flow will be the wet deciduous forest environment, while the vegetation type most resistant to gene flow will be the water environment, or the river. This is how we collected the genetic data. How we did it was we used larval tail clipping methods. The larvae were captured using dip nets, and a small 1 centimeter tail clipping was collected from 12 to 30 larvae from each pond. Tissues were stored in 95% ethanol. DNA extraction was then performed on the collected tissues. The resulting genotypic data was then analyzed using GeneMapper software, and pairwise FST values were derived. FST stands for Fixation Index and is a measure of genetic diversity. As far as the geographic data, the pairwise geographic distances were derived from the GPS coordinates from the ponds using the ArcMap program. Additionally, a raster surface detailing the 5 vegetation types was created using data from the United States Geological Survey. As you can see, there's a model that's on the slide. You can see that there are 50 white dots scattered throughout the map, and that represents the 50 sample ponds. The orange on the map represents areas of human influence. The blue represents the water, the aquatic environment. The light green represents the dry deciduous forest. The medium green represents the wet deciduous forest, and the dark green represents the coniferous forest environment. This resistance surface is a spatial GIS layer that represents the hypothesized relationships between landscape variables and gene flow that can be tested for statistical significance. For the computational methods, the pairwise FST matrix, the pairwise distance matrix, and the raster surface were analyzed using an R package called resistance GA. What resistance GA does is it iteratively assigns resistance values to each of the resistance categories. Instead of relying on expert opinion to parameterize the resistance surface, we will derive the resistance values computationally using resistance GA. It does this by making use of genetic algorithms to simulate hundreds of generations of gene flow for the study landscape. It also constructs 3 different statistical models to describe the genetic diversity: the null model, the distance model, and the vegetation model. This is a slide that depicts each of the 3 models that resistance GA will produce. The null model, as you can see, is the first model, and it has a lone explanatory variable that is PPID, and this is a random effect term that identifies each unique pairwise pond combination. The distance model is the second model, and it contains an additional explanatory variable in GEO. This is a fixed effect term that represents the pairwise geographic distance between the ponds, and the vegetation model further adds another explanatory variable termed land. This fixed effect term represents the landscape categories, and as you can see, each of the models has FST as its response variable. This is a scatter plot depicting the relationship between the log distance and the FST. And as you can see, there's a line of best fit on the plot. You can see that the slope of the line of the best fit for that plot is significant at a p-value of less than 0.001. And as you can see, the adjusted R-squared value is equal to 0.044, which represents that the 4.4% of the variance can be explained by the model, 4.4% of the variance of the FST value. This suggests that there's plenty of variation that's not explained by the geographic distance, and that the composition of the landscape is potentially important. Now for the model comparison slide, you have the 3 resolutions that we ran the models at on the on the side, and you can see that we ran the model at 50 meters, 75 meters, and 100 meters. 50 meters represents the finest resolution, and 100 meters represents the coarsest resolution. DF on the columns represents the degrees of freedom, or the number of parameters associated with the model. The AIC stands for the Akaike Information Criterion, as a measure of the quality of the model. AICCC is Akaike Information Criterion corrected, and is a corrected form of the AIC, of the regular AIC, and it's a safeguard against overfitting and penalizes the model for having too many parameters than it actually needs. The DICC is a delta AICC, and just tells you the difference between the best model based on the AICC and the model in question in that row. The natural log likelihood is the ln/lik, and that represents the log likelihood, the maximum likelihood point. Chi-square value was derived from that natural log likelihood value. The chi-square degrees of freedom is associated with the chi-square distribution, and the p-value represents the how the distance model relates to the null model. If it's in the distance row and it's in the vegetation row, it represents the significance of the vegetation model in relation to the distance model. So as you can see for the 50-meter resolution, you can see that the distance model is significant at a p-value of less than 0.001, and you can see that the vegetation model is not significant at a p-value of 0.735. That means the distance model is the best model at 50 meters. The same conclusion is true of the 75-meter. As you can see that the distance model has a p-value of less than 0.001, and the vegetation model is a not significant p-value of 0.91. However, you can see that in the 100-meter resolution, you can see that the vegetation model was significant at a p-value of 0.004. So whether I would probably trust the results of the two finer resolutions over the course of the one coarser resolution. And so based on these results, I would conclude that the distance model best summarizes the variation within FST. Now for the resistance values that were assigned to each habitat type, you can see that there are 5 habitat types in the very first column. And as you can see, we ran it at a 50-meter, 75-meter, and 100-meter resolution. And as you can see, while for each resolution, the resistance values varied slightly, you can see that the order in which they were in does not change. You can see that the wet deciduous forest was always the least resistant vegetation type at each resolution. You can see that coniferous forest was the second least. You can see that the dry deciduous forest was in the middle. You can see that the human influence was the second most resistant resolution, or second most resistant vegetation category, and you see that the water was always the most resistant category. As far as the conclusions that can be made from this, I would say that you can decide that the IBD model is better than the IBR model as far as explaining FST. You can also say that the wet deciduous forest was the least resistant vegetation type, while water was the most resistant vegetation type. While I did not predict that the distance model would fare better than the resistance model, the conclusions from the resistance model still support previously published literature regarding landscape genetics and amphibians. It's been previously asserted that rivers and anthropogenic practices have the potential to inhibit gene flow, and that's what we found. It is possible that the landscape of Mammoth Cave National Park is simply not heterogeneous enough for the variation in landscape features to significantly affect dispersal patterns. If you were to compare this landscape to one with more human influence, you might find that human influence is an even greater obstacle to gene flow than a river. But the findings at this scale indicate that this continuous habitat comprised of the 5 landscape categories has little effect. As far as these findings relating to amphibian conservation, I propose that limiting the creation of unnatural waterways, as well as reducing human influence in areas known to be amphibian habitats, could potentially lead to gene flow and increased diversity as a result. I would like to take this opportunity to thank the Honors College for allowing me to have this opportunity to present my thesis. I'd like to thank the WKU Biology Department, Biotech Center, Biodiversity Center for also allowing me to use our facilities in order to conduct my project. I'd like to thank my thesis committee, Dr. Jarrett Johnson, Dr. Scott Grubbs, and Dr. Doug McElroy for being a part of my committee. I'd like to thank Kevin Martin and Hilly Austin, who were students before me, whose work has been really helpful in creating my project. Again, thank you so much for your attention.